now that everybody has finished up their King of the Hill clip, um, let's start out by introductions in case anybody's new. My name is Amber Freed. I am executive director, and I wanted to take a minute for Becky to give all of us an update about her daughter Ella and what their family is going through.
Hi, everyone. I know a lot of you. So Ella was having some issues and she was having some new symptoms starting back in October. We really didn't know what was going on. But as it turns out, she was developing hydrocephalus. And we went to a neurosurgeon last week after having an MRI last month, and she had surgery. Actually, we went to the neurosurgeon on the 12th for the first time. So that's 2 weeks ago now. My weeks are kind of a blur. And she had surgery last week on that. She was able to have what's called an ETV, where they just make a new pathway for her cerebral spinal fluid to drain. She's doing really, really well. They did have to shave more hair than I thought they were going to shave off of her head. So we ordered her some little coverings for that, but she's doing really well. She's actually still sleeping right now. We're home from the hospital, but she is doing quite well. So we're very happy about that.
And how long does she have to be homebound?
So she's been like, do you mean after surgery? So she can't go out until after our follow-up. So that's on February 9th. So she's not allowed to go out in public and be around people. Of course, I'm sick because I must have picked something up in the hospital while she was in the hospital. So I don't think it matters too much because now I'm sick anyway. But she's not allowed to be around crowds until we get clearance from the neurosurgeon.
I hope
Now,
she feels better soon.
Thank you.
our hearts are with you, Becky.
Thank you.
And a reminder about Bo's podcast, it's exceptional. Is anybody else new here today and would like to introduce themselves? Okay.
Yeah, hi, Malte here. Nice to meet you all. I was very happy to be brought in touch with this really wonderful group through Irina. Very happy to have joined this club here. Looking forward to the talk today and to the discussions, and of course all the flavors that that USP7 has. Thank you.
Thanks for joining, Malte.
Amazing. And I am about to put a Google Sheet link into the chat because we're making an email roster so that if you have a question for one of your colleagues, you can easily contact them after the fact. But I'm not going to let it hold me up. So look for that in the chat box in a minute. So Emily, I'm going to stop sharing so that you can have the screen.
Hi. I didn't prepare any slides or anything because I didn't really know. But I got a grant, so thank you to the Hao-Fountain Syndrome community for that, and uplifting athletes. So it gets to fund future research to kind of expand on what I've been doing in my PhD, which has been focusing on Hao-Fountain Syndrome, and kind of go into a more cellular context of things, hopefully with fibroblasts, and just continue on the work I've been doing trying to understand MAGEL2 interactions with USP7 as well. So that's super exciting. Next weekend, or this weekend, I guess, I'm accepting the draft award, I guess, in Philadelphia. So, yeah.
And this event is so much fun. It's like an NFL grant, as Europeans may know, American football. So much energy, so much celebrating. It is like the Super Bowl of science. There's a lot of professional athletes there, and it is just a night to remember. I know Becky was planning on going, but can no longer go. So there will be a patient family there as well. Okay. Can everybody see my screen again?
Yes.
Alrighty, there actually are some funding opportunities right now through Simons and the American Epilepsy Society. And we also just applied to Cure Epilepsy to co-fund a grant with them as well. So Simons is pretty focused on autism research. They also have data and they're willing to share very easily. The American Epilepsy Society has a number of grants, and we co-fund those as well. So it's always worth applying to things, especially co-funding opportunities, because a lot of times, if an organization decides not to co-fund, they'll still ask us, do you want to fund this in part or in whole? And it's just an opportunity for more visibility and to get something funded. Those links will go out in the follow-up email. A big round of applause for new research. This was something we already knew about, but it is finally peer-reviewed. I don't think we have any of the authors on the call. And then turning our attention to 2026, we have lofty goals this year, and a lot are clinical. We are going to publish a disease concept study, a strategic research plan, and a standard of care. For this, we're going to need more people involved on the patient side of things. We want to analyze our data in Matrix, which is a large focus of this call, and February's call as well. We also want to have an FDA listening session so that as we bring drugs for clinical approval or repurposing or whatever that case may be, the FDA is already familiar with our disease. Scientific goals, I think we're finding that we really need to understand the mechanism of disease and complete more basic science in order for a small molecule screen to be successful. And we're moving that along. We have you guys to make this happen. And so our lab meetings, I'll touch on the February lab meeting in a couple of slides. We're going to spend more time thinking about omics in general and also take inventory of our existing models, but our wish list to make that basic science be successful. Everybody knows about our resources, and I'll send out these links afterward. We have a lot of resources, and now it's all in a Google Drive, so you just need to click and you can get whatever it is you wish. We have a lot of patient information in a resource called Matrix that Bo is going to talk about. And our symposium is going to be October 7th in Philadelphia. October 7th is going to be Scientific Day. October 8th will be Family Day. We encourage everybody to go. We will also have travel grants available. We want as many people there as humanly possible. In February, Combined Brain is going to be speaking about their integrated multi-omics project. This exceeds my education level. I do have it in email though. It's really going to build off of today's discussion, and I think the best way for me to talk about to give an overview of what the multi-omics project means is going to be in the follow-up conversation. But if you're interested in today, in February is actually putting more data to use, like running whole exome sequencing on the patient samples we already have held in the biorepository at Combined Brain, comparing them against healthy volunteers, and really start digging in to differences with those existing samples and building that biorepository even further. With that, we're going to start today with Dr. Benedikt Kessler and to discuss the bioinformatics of USP7, and I will let Dr. Kessler give his own introduction. Dr. Kessler, you're muted.
Yes, thank you very much. I'm just trying to share my screen. Voila. Okay. Yeah, it's something bizarre. You can just see white, snow white. It's like a snowstorm at the moment, just white, huh? Hold on. Okay, stop again. And then here. Oops. Oh, I see. No, it's actually okay. Right. Try again. Right, shared screen again. Up and up. Yeah, I'm just again trying. Hold on. Now it should work. I hope you can see the screen now. Yep, I can see some nodding. Okay, so first of all, thank you so much for the invitation, Amber and Bo and everyone, for this opportunity to give a presentation. And actually, I have been asked to sort of give a bioinformatics perspective to USP7, and previous discussion with Bo and with Amber clearly show that there's a need to dig more into data sets that are available also from the scientific community to contribute to understanding USP7. But I think it's important that multiple elements, people with different expertises are coming together, because one inherent problem in science is that each one has his or her own specific expertise, and they look at the problem in a specific way and don't necessarily come up with the right conclusions unless we take a step back and listen to all the different people with different disciplines to get a better picture here. As you can see here from this cartoon, no one from these scientists concluded that this is actually an elephant they're investigating this in this case here. So what I'm going to do in the talk today.
Benedikt, we're still seeing your first slide, or at least I am. So I don't
With the
see another-
cartoon. With the cartoon.
I see a
Can you see
building.
No, no cartoon, just the
Title.
title slide.
Okay. Then
you're-
I'll stop. Hold on.
You're-
Yes.
Be in the slide.
Yeah. Mm,
Yes, second slide.
is that now fine?
No,
Can you
it's
see
not.
it?
Now it's not in presenter mode. We just see the.
Yeah, hold on. I'm just going to put it back into presenter mode. Thank you, Tizio. That's good. Probably what I will do is to do this. Back over here. Can you see something now on the screen? What can you see now?
Pelican.
Oh, right, okay. Right,
Do you have two screens, Benedikt?
Yes, I have two screens, yes.
Sometimes I have trouble if I don't disconnect my second screen.
Yeah, I may have to do that. Can you see now?
Yeah, we do.
Yeah, now it works, huh? Yeah, well, I'll then take it out of presentation mode. Okay, good. And now you can see the cartoon as well?
Yeah. Yes,
Okay,
we do.
great. Good, yeah, good. Then let's get going. So what I will try to do is to cover a little bit of USP7 function and how actually different bioinformatics tools can be used to shed more light on its gene variant structure and function. So the USP7 is actually part of an enzyme class called ubiquitinases, as shown here. A subclass referred to as ubiquitin-specific proteases. And there are about 100 genes in the human genome that encode for these type of enzymes called DUBs. And most of them are involved in regulating protein turnover, as shown here in this cartoon here, where proteins are synthesized, and then when they come to the end of their lifespan, as you can see here, they're actually attached with different of these round balls here, which is actually ubiquitin, to form these polyubiquitin chains. And that is a signal for the cell to say, hey, this protein is no longer needed, no longer active, it needs to be degraded. And this polyubiquitin signal is then actually a way to direct it to this huge complex here called the proteasome complex, where then these ubiquitinated proteins are degraded into smaller pieces, peptides, and then by additional proteases back to amino acids. And this feeds then that back into protein synthesis to generate de novo new protein. So we call that the protein turnover cycle, where ubiquitin plays a key role, and DUBs, or like USP7, play a role in deubiquitinating these proteins here, and therefore affect these protein targets' lifespan. So that's what the fundamental biological role of DUBs are, including USP7. Proteins like USP7, they never work alone in the cell. So this is nicely shown here in this cartoon from this recent review here. Often Deubictinase enzymes like USP7 are in a complex and often with a enzyme that does the opposite. So USP7 or DUBs, they cleave ubiquitin from substrates, but they are in association with ubiquitin E3 ligase that actually put ubiquitin back onto substrates. So this means that there's a balance between an enzyme that adds ubiquitin, in this particular case here, an E3 ligase called MDM2, which is known to interact with USP7. And this adds ubiquitin to a protein called P53, which is an important tumor suppressor gene. And USP7 then cleaves ubiquitin off from P53, and that leads to its stabilization. So often in the biological context, enzymes with opposing actions are actually paired with each other, and then this allows the fine biological tuning of these activities either towards, in this case, more ubiquitination or more deubiquitination. This becomes important for the rest of the talk because USP7 not only interacts with this E3 ligase called MDM2, but also the other E3 ligases, in particular in the context of brain-related pathology. So again, because this is a talk about bioinformatics, what can we learn from bioinformatics tools or dig and mine data that exists. Here is the first one I would like to present. So one bioinformatics tool is called UniProt or SwissProt, which actually is a publicly available database dedicated to understanding protein function. So it actually contains information about most proteins expressed in the human genome or human proteome. And when you look at the entry for USP7, which is also called UBP7 or ubiquitin binding protease 7, you can actually you see that you can get a lot of information out of that. You can see that it has different names and it's also called herpes virus associated ubiquitin protease or ubiquitin thioesterase. So let's have a quick look a little bit what UniProt or SwissProt has in terms of USP7 related information. And as you can see here, there is a lot. So USP7 seems to be, at least from the basic science, one of the most studied DUBs at present. So you can see here function, so many different papers that have been looked at USP7 and identified potential processes where USP7 may play a role. And I highlighted here in red probably one of the most important ones, at least to date, where USP7 is thought to either deubiquitinate P53 or actually also MDM2 and regulate the actual P53 and MDM2 levels. This is very important because P53, which actually stands for a protein that has a size of 53 kilodalton, that's what P53 actually means, but it is a very important transcription factor, a tumor suppressor that controls cell growth. So if P53 is present, then cell control is restrained. But when P53 gets lost or is mutated, like in many cancer types, then the actual control of cell cycle progression is compromised and cells start to grow in an uncontrolled fashion, and that contributes to cancer development. But as you can see here, many more functions have been delineated and published, but you may also notice there's absolutely nothing about Hao-Fountain syndrome or associated pathways, at least in this entry as shown here. So let's go first a little bit into trying to understand what the biological function of USP7 is. And one way to do that is to investigate what USP7 interacts with in the cell. And As you can see here on the left, this is another entry information from UniProt, as you can see here from the website. This gives you this kind of table or this format where you have USP7 over here, and all these proteins here have been described to be in complex binding directly to USP7 or are in a complex with USP7. And I highlighted here the herpes, the Epstein-Barr virus nuclear antigens or viral proteins, also known to interact with USP7. And you find P53 as well here, as I indicated before in the slide. But on top of that, there are many, many more proteins which are reported to interact or be in the same complex as USP7. Since bioinformatics is the main topic of this presentation, so here the introduction of another tool called String. And this is again a publicly available a research tool or bioinformatics tool, or a web-based one that collects information from the literature, from evidences that are actually publicly available, papers and websites and other databases to build a network, an interaction network of USP7. And I just did that as a sort of illustration here on the right on this slide, showing that USP7, and then you see these lines, which indicates that the program or the algorithm actually found evidence for either direct interaction. So you can see here MDM2, that's the E3 ligase, direct interaction. This UBA52 and UBC, these are actually ubiquitin encoding genes, so it does interact with ubiquitin. But you see also other proteins like DNA, methyltransferase 1, or TP, a protein that binds 53. And this is possibly another E3 ligase that USP7 interacts with. So indicating that USP7 actually does many, many things in the cell. When you can then also check what is known about where it is. Where is it actually in the cell? And as you can see here again from the entry extracted from UniProt, SwissProt database, it's actually predominantly existing in cellular nucleus. And this is also described by numerous publications. So mostly, not entirely, but mostly present in the nucleus. So now it's actually very important to understand what USP7 does in normal physiology, so what its job actually is in normal healthy cells. But then comes the point of when things go wrong. So and often this has to do with USP7 somehow being derailed by its function. And one way to do that is by when it becomes mutated. And this is also quite nicely collated in this SwissProt or UniProt database, as shown here. So you have the disease variants linked to USP7. And there, to my delight, Hao-Fountain Syndrome actually comes out on the top. So this immediately lists what we call coding mutations. So mutations in the DNA, in the gene, encoding for the USP7 protein, that then leads to amino acid substitutions on the protein level. So you can see here methionine to isoleucine or glutamic acid to lysine residues. And this means that the composition of the amino acid sequence of USP7 becomes altered. And that, when it's not what we refer to as a conservative mutation or amino acid change, which means that the amino acids are looking chemically, they are quite similar. But as you can see here, some of these changes are chemically very distinct. So E, glutamic acid, has a negative charge, lysine has a positive charge. This means that it's quite logical or make sense to assume that these may then lead to structural alterations of the USP7 protein, and I'll show that a bit later in one of the later slides. So clearly some of these are non-conservative, and the prediction is that they may have consequences on USP7 structure, and then as a consequence, it may not be able to do its job as it should do under normal physiological conditions. So you can actually dig a bit deeper into this UniProt, SwissProt database, and you can get some more information about where these positions are in the protein sequence. So the protein sequence is actually here. That's this top, about 1,100 amino acids long. It's quite a long, a big protein. That gives you the positions of where these amino acids, these variations occur, and if there are any publications that are listed or available in the public database or public domain that indicate what the consequences of these variants actually, what they do. And again, most of them here are involved in more p53, MDM2. This is the major part that has been studied before, but now because of publications also from people on this call, and I'm going to come to that in a minute, we can see that Hao-Fountain syndrome associated variants are now coming up also in this database. So now the story is always much more complicated, and if you then go and ask, well, how many USP7 related pathogenic variants are actually known? You can see here from this slide, again from UniProt, SwissProt, that there are actually a lot. So you can see here again, this is plotted in function of the entire sequence, 1102 amino acids, and each red dot here will indicate a potential mutation that is considered likely pathogenic or somehow interfering with function. And there are ways to calculate that. So again, because of the amino acid chemistry and the change, and because we know a lot about the structure of USP7, one can predict and also calculate whether these amino acid changes will lead to substantial structural rearrangements or not. Now, the story is even more complicated because there are many more variants that have been described, and that is shown here. So this is the total picture of all variants at least currently described or documented in the UniProt, SwissProt database. So in red, we have the likely pathogenic ones. Then we have additional modifications or mutations that may not do anything, are likely benign, or and then some where we don't really know what the outcome is because this is all predicted. This is not experimentally verified. You can see here how many variants actually do exist, and a fraction of those are likely to be pathogenic. And there's a lot that we currently don't know about them, which then still await experimental validation. So, on top of that, I think to make you aware of additional resources from the bioinformatics side is to introduce the biobank, or what we call the UK Biobank, UKBB, which is a particular resource based on the voluntary take-up of about 500,000 donors, or people who decided to volunteer and provide blood samples for analysis. So 500,000, 500,000 people. And this is a great resource that allows much deeper phenotyping and also look at the genomic data. So exome sequencing has been done on these 500,000 samples from the UK Biobank. Also, metabolomics has been done on blood metabolomics on these samples, about 300,000 samples. And there is a big effort currently going on to also do blood proteomics on all 500,000 samples from the UK Biobank. And I think what is important in terms of as a resource is that most people who actually decide to participate and donate blood to participate in the UK Biobank are mostly healthy, at least relatively young, or mostly healthy people. And the idea is that we actually capture these, UK Biobank captures these donors or these individuals from the UK mostly early on before they catch disease. So for early disease detection, an absolute gold mine, because the idea is that then these individuals, these half a million individuals, will then be followed through their lives over decades, and then a certain percentage of these half a million people will then develop certain diseases, cancer, neurodegenerative diseases, other autoimmune diseases, and having samples very early on before they catch disease or at the very, very early onset of these diseases is a extremely valuable way to understand whether there are any molecular blood signatures in particular that would indicate disease progression very early. And why do we do that? Why is that so critical? Is that in particular for cancers, but possibly also for neurodegenerative diseases, is the earlier you can detect these diseases, the better or the more successful you may be able to treat them going forward. So I was very fortunate to have a very talented PhD student in the lab, Frederick Lassen, who actually worked a lot with this UK Biobank resource. So he was able to get access to this data. And we were trying to see whether in the DUB or USPs and also E3 ligases, how many variants can we see in the UK Biobank, and can we somehow link some of these to existing human disease conditions. And the good news is that for most DUBs and also E3s, there's a lot of variants that do exist. But the challenge is then to actually then try to see whether these variants, genetic variants in particular, are then really disease causing associated to disease, or whether they are actually just a reflection of normal variation, but are actually benign and just part of diversity, but all physiologically. So this is actually not easy to discriminate, to find amongst this wealth of different mutations and different variants, which ones are really disease associated or causing disease, and which ones are just bystanders, and which ones have actually nothing to do with disease progression. So in order to answer that question, one then needs longitudinal data sets also on the state of health of these individuals who gave the blood, to then associate their genetic profile with disease progression. So I understand that there is an appetite maybe to interrogate the UK Biobank a little bit also for USP7 gene variants and see whether that can capture other additional type of USP7 associated diseases, or maybe even linked to disease pathology close to the Hao-Fountain syndrome. So, but there are also other databases, and I would like to, and bioinformatics tools, I would like to mention particularly this one. There are many more, I'm aware of that, but in the interest of time, I can only mention a couple of them. But I would like to mention this one called DUB-ACE or DUB-BASE that was described about 5 years ago originally. And the reason why I would like to highlight this one, as shown here, so this is actually, again, it's a web-based bioinformatics tool, so you don't have to pay anything. It's not behind a paywall. You can just type in the URL down here, and you get directly access to it without any restrictions. Interestingly, it allows you to look at DUBs and their associated potential substrates, again, based on public data knowledge. And it can also allow you to look for clinical variant mutations and see how whether some of them may be linked to a certain disease phenotype. And I like that example because here the search term was related to impaired brain development or brain function, so autism, attention deficit, mental retardation, microcephaly, and so forth, and ask whether there are any DUB-related genes associated with these clinical variants. And the answer is yes, a very strong yes, there are. And if you go through that workflow, you actually get, for some of the DUBs, including USP11, USP9X, but interestingly, for this talk here, USP7, at least that database claims to associate quite a few of these clinical variations to neurodevelopmental issues. So in light blue, links to neurodevelopmental properties. In green, dark green, intellectual or disabilities, and in light green, microcephaly. And not only it gives you these variants, but also gives you the genes or proteins that somehow are linked to that. So this is, to my delight, one first, or probably one of the first bioinformatics-based predictions. It's not the only one. I'm absolutely aware that it's not the only one, but it lists some interesting genes linked to USP7 that somehow have clinical variants associated with them in the brain pathology space. And we can see some other E3 ligases. We can see other type of proteins. So I think this is a ideal hypothesis generator, which allows us experimentalists to then really go and find out functionally what is that connection, and how does that then really affect brain function or neuronal function in sort of biological experiments. So at that stage, the questions are, what can we really harness USP7 based research knowledge based on bioinformatics analysis of what is actually available to date, and how can we create new medicine out of that? So I think this is possibly a long-term goal of this organization here, the Hao-Fountain Syndrome Foundation. And certainly for us, researchers who have done a lot of basic research on USP7, is actually an interesting way to create more impact on the research that we're doing. So what was discussed also in previous calls and today is to try to manipulate USP7 function using small molecules. If you want to reduce, if it's too active, you're going to use inhibitors to reduce activity, or if the mutations are interfering with functions, you try to restore functions by using small molecule activators. The other possibility is gene therapy. It's trying to replace a defective USP7 gene with a correct one, and therefore restore function. I think for the rest of the presentation, I would like to focus on the first part, so small molecule development around USP7 that could then correct USP7 function to a more physiological level. And one is actually more than 10 years ago, about 15 years ago, where at that time USP7 was clearly defined as a key enzyme that does stabilize or control stabilization of this tumor suppressor gene p53, which is very important because it's either p53 is either impaired or even lost completely in about half of all the cancers. So the therapeutic window would then be to try to see whether a small molecule could then lead to increasing the stability of the p53. And the way to do that is then to interfere with the ligase MDM2 that ubiquitinates p53 and again leads to its degradation by the proteasome. And because USP7 also deubiquitinates MDM2, the E3 ligase, it can control MDM2 levels. So when you inhibit USP7, MDM2 is auto-ubiquitinated, is then itself degraded, can no longer ubiquitinate p53, and therefore lead to stabilization of p53. So that's the hallmark of the clinical rationale to develop inhibitors. And this is usually not done sort of in a, well, I have to be careful because Sarah Burlake is actually on the call here. I saw her, at least in the call. Chemistry is done in some of the labs, but this is usually a big effort to come up with small molecule inhibitors. Can be done in academic setting, but this is also what industry is good at. And at that time, we formed an alliance with an American company in Boston, Forma Therapeutics, with a number of academic labs. Some of you may recognize some of these personalities here, who actually would be nice to invite, maybe also to give a talk in the USP7 virtual labs. They will have nice stories to tell in relation to USP7, but also DUB biology. David Commander, Sylvie Urbe, Mike Clegg, Hype Bova, who unfortunately has passed away, but then also Catherine and Stefanos from Forva. And together, doing a small molecule screening campaign, we identified 2 molecules, FT671, so FT means Forma Therapeutics, number 671, or Forma Therapeutics 807. So that's a covalent inhibitor, that's a non-covalent inhibitor of USP7. And at that time, it was, people were very doubtful that you can actually get very selective inhibitors. So Adan Pinto-Fernandez is actually also on the call today. At that time, was very keen to test them in a cellular system that we have using activity-based probe labeling. So in this case, you would take the compounds, incubate them with cells or cell extracts, create extracts, and then you can use this ubiquitin-based probe to label cellular DUBs. And if a compound has engaged with a particular DUB that competes with the probe, with this ubiquitin probe, and you can visualize that by Western blot. So here you can see no probe, that's the band of the DUB. When you add the ubiquitin probe, adds about 10 kilodaltons, you can see that shift. And then when there is target engagement of these compounds that you want to test to that DUB, then the probe can no longer bind. So you see that collapse back to the original molecular weight of the DUB by Western blot. And that's exactly what Adan did for FT671. You can see here, exactly like the cartoon, you can see the probe adduct. And when you add compound in increasing concentration, that goes down again. You can see there's interference of compound binding to the catalytic site so that the probe can no longer do it. And this happens in extracts with both compounds, 671, 827, but also in cells. You can see this competition, which is for us a clear evidence for target engagement. These compounds bind USP7. Also, together with Andrew Turnbull, collaborator from Cancer Research Horizon in London, we were able to get this crystal structure of USP7. You can see that here with the catalytic site down here. And when ubiquitin binds, you can see that orange blob here that binds to the catalytic site. And this is exactly where the compound actually binds. You can see here FT671 and FT827, they bind to in the region of the catalytic site and in an area where they clash, where the C-terminal part of ubiquitin is usually binding, so therefore preventing ubiquitin from binding to the enzymes. This is how it leads to inhibition. And to our amazement, this seems to be quite selective for USP7 because it's only USP7 that has enough space to accommodate that inhibitor. It's shown here in yellow in this cleft close to the catalytic site because these switching loops are sort of pushed away. And if you compare with other DUBs, Yes.
I am so sorry to interrupt, but we have 10 minutes. And Bo, I was hoping um
Okay.
to spend a couple minutes on what we have in
Good.
Matrix. Yeah, and because this is going to feed into the February meeting, and this is going to be such a great discussion.
Yeah.
And it's
Yeah.
the
Yeah.
best thing when one meeting is so cool that it actually takes two meetings. So, Bo, can you give an overview of what we have so that we can talk as a group about our omics project?
Of course. Do you
Yeah.
want me to share my screen? You want me to show you?
Yeah. Then I
Yeah.
stop sharing my screen. I stop sharing mine. Yeah, that's fine. Yeah. Good. So over to you, Bo.
Thank you. Okay, so just very informally, what I've been doing is collecting data from Matrix. That's the platform where we keep our patient registry, and people have been fairly good about uploading full medical records from all their hospital visits and all their doctor visits, but also genetic reports. And so what we've tried to do is just assemble some genotype and phenotype data based on those records. So what you're going to see is really not anecdotal. It's all from documents that people have uploaded for the most part. So what I wanted you to see first is just the phenotype data. What we've done here is just kind of define our disease based on the phenotypes that we're seeing the most. And so you see I'm trying to use HPO labels. And so there's two tiers of these. This is the first tier up top is kind of what we see in most cases, and then tier two is less common. But tier one, those are kind of the core features, and then so you'll see the patient ID here on the left, and which of those phenotypes are visible in those patients, and then let's see. I'm going to stop sharing for a second. And I just want to show you the genotype data now. Here we go. Okay. So can everybody see that? So a few things I just want to mention about this. So we have roughly 80 or so patients for whom we have the genotype data. So a few things about this. One, we know of at least two sets of patients who have the identical variant. And so that's interesting to us in the sense that we are in touch with some of those families, and we know just anecdotally sort of what the phenotypes are, but unfortunately not all of them cross over into the realm of having the actual documents and the phenotyping data. So we have some work to do there. But I think exploring those four patients at least and kind of seeing what those phenotypes look like when we know that they have the same variant is interesting to us. You'll also see from this table that we have several individuals for whom there are multiple variants aside from USP7. So that's something we need to think about, figure out how to account for that. But mostly what I think we need here is a lot of help, and that's in talking about what you see here as unknown. You'll see there's a lot of spaces on here where we have unknown, and a lot of that is just a need for us to account for different types of missingness. Like sometimes when it says unknown, it's because the lab report didn't say so, or they didn't test for it, or we have only a partial lab report and we're missing a page, or there's a lot of different reasons that something is unknown. And so I think we really need help in refining this table and making it better so that we don't just say unknown, 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 but we say what type of unknown it is, and thus it becomes useful. And so really, our goals with this data, number one is we really want it to be more complete. We do have a little over 300 patients in the world so far that we know of, and we have only a fraction of the data for them. And we're really working to motivate our group and try to get them to provide more of their data. But we want it to be more complete. And aside from that, we want it to be a cleaner data set. We want to standardize it. You'll see here, I don't have a scientific background. I don't have a genetics background. So I'm going based on what's in the reports, and you'll see I need some help here in making it more uniform and making it standardized so it's useful. And so the first goal is a complete and clean data set. And the second one, I think, really ties in well, Dr. Kessler, with what you were talking about today. I mean, when you showed the features of UniProt, that blew me away because, we're really interested in finding out about what molecular tools are out there to show us what are the consequences of a given mutation. What impact does that have on activity and ubiquitin tags and what cell pathways are destabilized? And so when you showed that to me, that blew me away. So we would really love to combine our data with those molecular tools to the extent we can and map some of the molecular signatures to the clinical signatures that we have. So I'll stop there and pause for questions or comments.
I can add
Actually,
something about February and the meeting, how we're building on this and the combined brain omics data project that we have the opportunity to participate in. And I'm just going to read it. There's 16 USP7 participants with paired whole blood and plasma in this repository. I would suggest running whole genome sequencing and a soma scan with the analysis, and it talks about cause. Then what we can do is enter this information into matrix, and the proteomics cases plus controls could aim for a preliminary plasma proteomics signature of USP7, pathway enrichment consistent with USP7's role in protein regulation, variant class stratification, showing whether biology differs by mechanism, and possible clinical correlation framed as exploratory. And this is a pretty low price because CZI, the Chan Zuckerberg initiative, is actually funding the majority of it for rare disease groups. So this is where the conversation is going to lead for February.
that's nice. It's just what I wanted to say, maybe in one or two more slides, is that Irina and Sarah actually have already some functional data on some of these variants, which is, I think, the over next slide that I wanted to show. But in the interest of time, maybe that could be presented maybe on another time. So there is certainly in Irina's lab and Sarah's lab, there are efforts going on to better understand what these variants actually do functionally. What is the consequence of these mutations? And then maybe if new ones come out through patient-derived sequencing, I think Irina and Sarah's labs have ways to better understand how USP7 function is impaired by these patient-derived variants.
Yes, and some of this project that I just read to you, because it's what SomaScan does, is overlapping. And so my thought is the virtual lab meeting is the second Tuesday of every month, and I would like it to be 9 a.m. Central because that works a little easier for some of our European folks to join. And so let me know about the time, guys. But we're leaving this meeting with so many cliffhangers. And February, we could leave this much more open to discussion with that proteomics person on the phone and Dr. Kessler, and spend half the time of just hashing this out. Does that sound good?
Sounds great. Yeah, I could then maybe show these slides in that context for next time. That might be actually quite useful.
That would be fantastic. And the other cliffhanger I'm leaving everybody with is that we have some cells from Charles River that we're about to get shipped out to various places. And we just need to finalize that. So there will be a flurry of emails regarding that too. But otherwise, you guys, we have so many tailwinds. This is so exciting.
Thanks, Amber.
Yep. Thanks, Amber. Thanks, Benedikt. Look forward to chatting again next month.
And
Yep.
Emily,
Yeah.
have so much fun at the draft. You better show up with pictures.
I will. Sounds good. Thank you.
Yeah, congrats, Emily.
Thank you.
To be
All right.
continued. To be continued. Yeah. Okay. Bye for now.
Bye.
Bye.
Bye, everyone. Nice to see you all. See you next month.